Hello, welcome to our problem session today. Today we'll be talking about biochemical pathways and mutation. Let's start with a biochemical pathway problem. Now, in your lecture, we did write biochemical pathways based on the data. However, today we're going to analyze a pathway looking at what would restore particular mutants to the wild-type form. Here we have a biochemical pathway. The normal non-mutant strains can produce the end product here. Mutant strains are shown over the arrow for which that strain is defective. So strain 1 cannot convert metabolite C to metabolite A. And then, of course, the metabolites and intermediates on the path are between the arrows. So which metabolite will restore the phenotype for strain 1? The rule for this is that metabolites that occur after the blocked enzymatic step can restore the path and produce the wild type. So if this step right here is blocked, metabolite E and F are the only ones that would be in the direct line after that path and could therefore restore the path. So if I provided strain 1 with metabolite E, it already has the enzyme to combine E and D to make F, and it already has the enzyme to convert F to the end product. Similarly, if I provided strain 1 with metabolite F, it already has the enzyme to convert F to the end product. Now it says, suppose strain 2 can be rescued by providing metabolites B, D, and F, but not the others. That means strain 2 must be in an arrow that's before all of B, D, and F. So let's see, there's B, D, and F. So the arrow that's before all of these would be right here. And strain 2 would be right here on the pathway, indicating it cannot get to the point where it can create metabolite B. Okay, the next part of this page has a coding dictionary, and that's for reference later on because we will need it when we look at our mutation problems. Let me see if I can sort of shrink it down so I can get to it later. The rest of our problems today involve mutation. It's important that you feel fluent with the vocabulary involved with mutation and that you can work problems comparable to these on this worksheet and in your homework in the various practice problems. A codon that specifies the amino acid glycine undergoes a single base pair substitution that results in a nonsense mutation. So we've got some vocabulary here. So a nonsense mutation is just a mutation that causes a stop codon to be put into that position. So it basically changes a codon that would normally code for amino acid to a stop codon. So normally the codon codes for an amino acid, it converts it to a stop. A single base pair substitution, that means we're changing one base for another, and that is a point mutation. A point mutation is defined as the substitution of one base pair for another in the DNA. A frameshift mutation is an insertion or a removal of one or more base pairs from the DNA. Okay, let's see. A couple of other things. What is a transition, and what is a transversion? So in this, you have to go back to remember which bases are purines and which bases are pyrimidines. A transition is like to like. So one purine is changed and substituted into the other purine, or one pyrimidine is substituted to the other pyrimidine. Now, when we're doing this at a DNA level, we're actually changing a base pair. So for a transition that went effectively from C, the pyrimidine to the other pyrimidine is T. then we're talking about a CG base pair being changed to a TA base pair. Versus a transversion, that C could change into either an adenine or a guanine, one of the other types of molecules, because C is a pyrimidine and adenine and guanine are the purines. So I'm just going to change it to an A. But that means at the DNA level, when we're looking at a base pair, we've gone from a CG base pair to a TA base pair. It's very important that you keep these in order so that here in the transition, the C changed to a T. Here in the transversion, the C changed to an A. Now, bringing this to our glycine. Here we notice that the amino acid glycine went to a nonsense mutation. If we look in our coding dictionary, glycine is here. GG anything codes for glycine. And then it went to a stop codon. So UGA, UAA, and UAG. So let's take a look and see how those work. We needed GG anything on the DNA to change to, well, we'll look at it in codon lingo. So I'm going to write a U here, even though the change would be at the DNA level. But our stop codons are UAA, UAG, and UGA. Since this anything position can be anything, it doesn't matter which of those it is. We need to only change one position, though, so we need to go to the stop codon that had the guanine in the middle, and that means this first guanine changed to the uracil. So on the DNA level, we had a G that changed to T, which means our GC pair changed to a TA pair. Remember, keeping the order the same, and that would be a transversion because guanine is a purine and thymine is a pyrimidine. So this is a transversion. A couple of more vocabulary words here. Another word for a silent mutation is a blank mutation. Well, first, what's a silent mutation? A silent mutation means that we changed the codon, but we did not change the amino acid. And this is another word for a synonymous. And then a missense mutation, we changed the codon and it changed it to another codon that coded for a different amino acid, but not a stop codon, because remember, changing to a stop codon is nonsense. So when we have a missense mutation, that is also called a non-synonymous. Did I do this right? Non-synonymous. There we go. I can't spell today. I think that one's correct now. Okay, let's take a look at another problem. So this one says we have a mutation that causes an addition, alteration, or deletion of one base pair, resulting in the production of a non-functional protein. So in this case, we've gone from a normal protein that had, looks like 5 amino acids in it, to something that was a lot shorter. When we see something like this that is changing multiple codons, because effectively we had to change this codon and then we had to have a stop codon there, you can just mentally put a stop codon there. Odds are this is going to be a frameshift mutation and not a point mutation. And it says it is either an alteration, an addition, or a deletion. So the alteration would be the point mutation. So these two would be our options for the frameshift mutation. And we need to figure out which one it is. So this is a little bit hairy. And what I like to do is write out the normal and the mutant protein sequences, normal. So methionine, histidine, ASPGLYRP. And I'm going to leave a space here because I'm going to write out the messenger RNA for the normal. And then I'm going to have the messenger RNA for the mutant, and then I'm going to have the mutant protein. And I do this so that when I'm looking at these two RNA sequences, they're right next to each other, so it's easy for them to track. So let's see, methionine, GLN for glutamine, and then we know that next had to be a stop codon. And I'm putting that in, even though it's not an amino acid, to remind me to write the stop codons down when I write out the RNA sequences. So methionine, let's see. AUG is methionine. Histidine, CA, either U or C. And I usually write my options like that. Asparagine, GA, either U or C. Glycine, GG, anything, and tryptophan is UGG. Now, methionine again, AUG. Glutamine, it's CA, either A or G. And then a stop codon, and we could have UGA, UAA, or UAG. And now I want to compare these and see if I can figure out where I maybe changed something. So yes, I could have changed CAU or C to CAA or G, and that would have caused the change in the amino acid, but that wouldn't have made the stop. So it's not going to be a point mutation. But let's see if I can look at some similarities here. If I inserted something right here for this position, inserted something right in there, then UGA would be my stop codon and would be in the next reading frame. So if I inserted something right there, then that would have done it. So in this particular case, I could say I inserted the AT, or I could say I inserted the GC, either one, but that insertion would allow me to get on frame. And so we will call this a frame shift mutation. It's an insertion. And the stop codon used in the mutant protein must be UGA. And the thing that I inserted was either the A or the G at this position, so either an AT pair or a GC pair. Okay. Let's look at this next one. Got a coding strand and a normal coding strand of a normal and a mutant sequence are both shown here. So both of these are the coding strands, which means we can mentally think of these as RNAs by changing the T's to U's. And then we want to figure out where the change is and describe this as either a point or a frame shift mutation and what changed. So if I look along these, there's something that changed. Yep, looks like that's the only change. So what happened is that this is a point mutation. And this went from a C to a T, which means it went from a CG base pair to a TA base pair, which is going to be a transition. Give the one-letter abbreviations of the amino acids in the normal sequence. Write only the abbreviations for the amino acid from amino to carboxyl. And if this was Moodle, don't put any spaces so that Moodle will grade it correctly. And that's really important. Moodle is incredibly picky because it will grade however it was programmed to grade, and it doesn't allow for anything that's different. So here we want the one-letter abbreviations for the normal sequence. We read along 5' to 3' until we find an AUG. Since this is the coding strand of DNA instead of messenger RNA, we look for an ATG. And then we split this into codons. And this is our stop codon. So we go back to our coding dictionary. ATG, first is going to be M for methionine. GCC, GC, C, looks like A for alanine. AAT, which would be like AAU, looks like N. GAC, G, A, C, D. And GAC, UAC, since it says T, remember we're going to mentally change that to U. So UAC, UAC is tyrosine. So our first initial protein spells a word, Mandy. Now you will note that some of these do spell words, and these words will all start with M because methionine is going to be the first amino acid in the protein. Now we want to do the same thing for the mutant sequence. Still AUG, still GCC, still AAU. So MAN is still the same. Now GAU, GAU. It still codes for a D. And then this last one, UAC, was the same as before. It codes for the same thing. Look at that. So when we think about the change at the protein level, even though we changed the codon, we didn't change the amino acid. So this is a synonymous mutation. And again, reviewing this vocabulary, synonymous, we change the codon but don't change the amino acid. Non-synonymous, we change the codon and it codes for a different amino acid. A nonsense, we change something that was a codon coding for an amino acid to a stop codon. This is going to result in a short protein. And then a read-through is opposite of that. We change something that coded for a stop codon into something that codes for an amino acid, so we end up with a longer protein. So a nonsense will give us a short protein, and a read-through will give us a long protein. This one changes the protein, but the same length. And this one, no change in the protein. Like I said, there's quite a bit of vocabulary involved with this, and I encourage you to make a list of it and then practice using the vocabulary as you solve the problem so that you become comfortable with it. I think we've got another problem or two here. Here we've got the coding and strand of a normal and mutant are given. So again, coding strand of DNA, so you can mentally change T's to U's to make it read like messenger RNA. We want to decide if it is a point or a frame shift, and then if it's a point, we'll talk about transition or transversion. If it's a frame shift, we'll talk about is it an insertion or a deletion. So let's read along. G, G, A, C, G. Okay, we got an extra A. So somehow we had an extra A, and then we're back on track again. So we inserted that AT pair. So it looks like an A here, but it would be an AT pair in DNA, and this would be a frame shift mutation. And it was also an insertion because we added that extra base. Give the one-letter abbreviations for the protein from the normal sequence, and then again, you're writing just the letters, one-letter abbreviation from amino to carboxyl end, no spaces if you're doing this on Moodle. AUG would be methionine. And that's the stop. So let's see what our coding dictionary has. We know methionine is AUG, ACU is threonine, AAU, asparagine, and GGC, G. And that's it. So MTNG. Sorry, didn't spell a word this time. Give the one-letter abbreviation for the amino acids of the protein from the mutant sequence. Okay, so start. 2, 3. Notice that we've messed up our stop. But there's one. So this one's going to be quite a bit longer. And I'm going to solve it with the coding dictionary right here so you can see that. AUG would be our RNA version of this coding strand, so methionine. ACT, which is like ACU, which is threonine. AAA should be asparagine. Oops, no, AAA. What am I thinking? A, there it is, lysine. Okay. UGG, tryptophan W. CUG, leucine. ACA, threonine. GCA is alanine. So hopefully we've got this now as MTKWLTA. And again, didn't spell a word. So at the protein level, this can best be described as what? It's not silent. We definitely changed that one. It's not missense because we didn't just substitute an amino acid. We did not stop short. We went long. So this one is a read through. Now this next one is a little bit tricky. It just requires, it's not any harder, but it just requires perhaps a few more steps because we have an initial protein, a mutant protein due to a single base substitution. So not an addition, but a substitution. And then we have an intragenic suppressor. So something else went wrong to change it to get it at least back on the right reading frame. So notice that we started off met lysine, CR ... met lysine, it stopped early. So this next one had to be a stop codon. And then somehow we got back on met lysine. This is still messed up, but then we're back on track. So an intragenic suppressor is within the gene, something changes to kind of get things back on track. Maybe not perfectly, but at least in this case, it was the same length and it only messed up one amino acid. So it mostly got it back. So let's see how that could happen. So if we start off methionine, AUG, lysine, the AA, either A or G. Now, serine, there are a whole bunch of them because there's UC anything, and there's also AG, either U or C. So that's one you have to be careful of. UC anything and AG, either U or C. And proline CC anything. Now, I'm not going to write the rest of it. There's nothing wrong with writing the rest of it. I'm just going to save a little bit of time because I know that my mistake happened somewhere in here, and my other mistake that got us back on track also happened somewhere in there. So met, AUG, lysine, either A or G, and then a stop codon, UAA, UGA, and UAG. It's one of those three. So what I'm thinking here is the codon for serine changed to one of those, and it said a single base substitution. So I want to look for one that's close. UC anything or AG something. So this AG, U or C, I'd have to change more than one place. So I must have started off with this UC, and that C must have changed to a G. So what I'm thinking is my serine codon was UCA. And I'm saying it's A because my stop codon had an A, went to a stop codon of UGA. So C to G. Now let's see if I can get this to get back on track. So methionine is still going to be the same, AUG. Lysine is still going to be the same, either A or G. Now, what is cysteine? Cysteine is UGU or UGC. So if that was UG, then it went to cysteine, UG, either U or C. And we can't tell which one it went to. It went to either one, either UGU or UGC. So the location and nature of the original one, we started off with a serine codon of UCA, was altered to the stop codon UGA, which was then altered to either codon for cysteine. The original mutation from C to G, which means it was at the DNA a there we go, CG to a GC. And the suppressor mutation, oh, and by the way, this would be a transversion because the C went to a G, and those are different types of molecules. The suppressor mutation went from a to either U or C. So no matter what, it's a purine going to a pyrimidine. So it's also a transversion from, we will say, an AT pair to either a CG pair or a UG pair. So UA pair, or a transition from AT to CG, depending on which of the codons we had for cysteine. So there's quite a bit in that problem, and it requires you to put together several things that we talked about so far. I think I've got one problem left. With multiple parts. So this problem has several mutations based on mutations of this normal sequence. And I suspect we'll be classifying the different ones as to what they are. So let me see if I can get started on that. There. So the normal sequence of the coding strand again, and several mutant sequences are below. Assume the alterations in the protein sequence alter protein function. So that means if we got a change in the protein function sequence, we actually got a change in the function as well. Classify each as a transition, transversion, frame shift, insertion, or frame shift deletion, and then describe the effects the mutation will have on the protein. So we'll have to kind of do a bit of translating as well to figure out what the protein effect will be. So let's compare this first one. A, 3 C's, TG. Whoa, there's our change right there. Look at that. We went from a CG pair to a GC at the DNA level, GC. and that's going to be a transversion. And if we think about what happened at the protein level, looks like that codon remained the same. This codon changed, so I'm going to just start translating the first part of this. AUG is methionine. ACT, which is like ACU. ACU is threonine. And this is, again, methionine. AGU is serine. So at the protein level, we switched, we substituted a serine where there used to be a threonine. So a threonine changed to a serine at the protein level. Okay, one down. Now let's see what we can do with number B. C, 3 A's, A, T, G, ACT, 3 A's. Huh, 3 A's, 2 A's. Got an extra A. Okay. So this looks like it's an insertion, doesn't it? That's my stop here. I'm going to go ahead and translate the rest of my initial protein. AAU, asparagine, GGC, glycine, and then it quit. So MTNG. So let's see on this one. I went from M, ACU is still going to be threonine. Now AAA. There it is, lysine, which is K, UGG, W. We haven't stopped yet, though. Gotta keep going. Finally, it stops. So this one's going to be a read through. CUG, leucine, ACA, threonine, GCA, alanine. So that went really long. So at the DNA level for mutation B, we had an insertion of that AT pair, base pair, which means our coding strand got the A, the template strand got the T. So it looks like there's just an A added here. So that's at the DNA level. And the protein level, we went from, it's a read through. So the sequence went M, T, N, G, went to M, T, K, W, L. So it's a much longer protein. And then let's see, our last one, go back and compare it to the very first one, C3A's TG, got that. ACT looks good. AAT looks good. GGC looks good. TGO, A to G. So we went from an AT to a GC. Not only that, this messed up our stop codon to something that codes for UGG, codes for tryptophan. So this will also be a read-through. Because now this codes for tryptophan, and then we've got, it keeps going. And it looks like we won't have a stop codon in the right reading frame. But we do have a read-through there. I think that finally worked through the problems I have today. I hope this has been helpful and that you feel like you've learned something. Thank you for your attention. Please contact me if you have any questions at all. Have a great day.